Hello, I am Sven Gudmund Hinderaker, professor at Center for International Health, University of Bergen. In this lecture, I will tell you about how drug resistance develops in tuberculosis. How can antibiotics kill bacteria? An antibiotic generally blocks a specific target in the bacteria and interrupts the bacterial life in this way. Here are some examples of targets for antibiotics. Protein synthesis may be blocked. DNA or RNA synthesis may be blocked. Cell wall synthesis may be stopped. Cell membrane function may be interfered with. Metabolic pathways may be blocked. This paper gives a good outline of the mechanism of drug resistance in Mycobacterium tuberculosis, and this table gives the gene involved and its function and the mechanism for drug action. As an example, we will look at the drug rifampicin, the best antibiotic against tuberculosis. Inside the bacteria, the enzyme RNA polymerase produces messenger RNA that is used by ribosomes to produce all the proteins that are needed by the bacteria. If the drug rifampicin is present, it will bind to the RNA polymerase in the bacteria and hence blocks the function of the enzyme. Bacteria have genes coding for RNA polymerase. The beta unit of this enzyme is RPOB. A mutation here does not destroy the function of RNA polymerase, but the drug rifampicin fails to bind or stick to it, and hence has no effect on these mutant bacteria. These mutants are very few, less than 1 in a million in a normal bacterial population. But when rifampicin is present, the normal bacteria are killed and the mutants survive and take over the bacterial population. In short, rifampicin selects the mutants. A medium-sized lung cavity in a patient with tuberculosis may contain 100 million bacteria. In this big population, a few mutant-resistant bacteria will exist to almost any drug. This is a list of frequency of mutations occurring in genes coding for various targets and common anti-tuberculosis drugs. The development of drug resistance is very dependent on a large population of bacteria and is basically natural selection of surviving mutant strains. After adding one antibiotic for a few weeks, you have selected a new colony of bacteria with millions of bacteria all resistant to that drug. If you now add another single antibiotic, the same selection will happen. A new resistant mutant will survive in the new environment with 2 drugs, and after a few weeks, this will be the dominant strain. So, which situations in a tuberculosis patient favor the development of drug resistance? Basically, the more bacteria, the more likely development of drug resistance. For example, the number of bacteria found in pulmonary tuberculosis is higher than in extrapulmonary. Pulmonary cavities contain particularly large populations of bacteria. The bacterial count by smear microscopy gives a hint of the bacterial load. Also, HIV-positive cases may contain huge numbers of bacteria with few signs. This has led to important policy implications for tuberculosis control. Some are listed here. Drug regimens are based on the expected bacterial load in the patient. Never treat tuberculosis with only one drug, unless you're sure the bacteria are very few, as in latent tuberculosis. Start treatment with 4 drugs when the bacterial population is large, in case there is initial resistance to some of the drugs. After 2 months, the bacteria remaining will be very few, and hence few drugs are needed to complete the treatment. Patients who have been treated before must always have a stronger regimen, as in some cases drug resistance has developed, and therefore drug resistance and susceptibility should be tested. The development of a new large colony of resistant strains takes a month or so. This is why a patient lost to follow-up for 2 months or more is regarded as treated before and needs a stronger regimen. How can we know about drug resistance in a country? There are some indicators that may suggest that drug resistance may be common. A tuberculosis program always reports treatment outcomes, and if the proportion of treatment failures is high, this may indicate resistance to drugs. If the proportion of patients lost to follow-up is high, you know that the program has big challenges in completing the treatment, and this may be associated with drug resistance. If the tuberculosis program is not ensuring drug taking by direct observation, there is a much higher risk of drug resistance. A tuberculosis program needs not test resistance of all patients detected, but should do drug susceptibility testing of all retreatment cases. A tuberculosis program should also consider regular national surveys of drug resistance. In conclusion, drug resistance will develop in a big population of bacteria when a drug is given. Drug resistance can be prevented by treating tuberculosis patients with multiple active drugs for the time prescribed.